Good morning, everyone. So my name is Yousri Martin. I am working at the Luxembourg Institute of Science and Technology in Luxembourg. In my team, we are working with traditional monitoring, biodiversity monitoring approaches, and also we are starting to develop more and more technological solution. And we will present two devices from my colleague in the afternoon. But here I want to talk to you about a new idea we had some years ago, which is actually using chemical attractant to attract insects and butterflies under the camera trap. So we took the opportunity of funding from the ministry because they wanted us to monitor some rare species in dry grassland area in Luxembourg. And to increase the chance to detect those species which are very low abundance, we decided to try some attractants. And we had only not a lot of money at that time, but 1 year to test something. So we actually built a prototype of camera trap, under which we presented some containers containing actually different attractants. So the containers were like a basic one with a wick passing through the cap. The cap were colored by special painting that actually mimicked the UV behavior of flowers on the field. And we had several treatments of attractants. So we had the Buglea davidi solution, which was made by our colleague at least. So we are working in a very big research center with more than 800 researchers in a lot of different fields, and the team is specialized in organic chemistry. So with them, we identify the most important chemical compounds that attract pollinators to this Buglea davidi, so butterfly bush flowers, and we put them in solution. We had another solution based on the Thymus serpulum, and we each time decided to add, we had also a control solution, so it was only water. And we decided each time for each place it will be one olfactive solution, so Thymus, control, or Buglea, and we added or not the nutritive solution every time. So the nutritive solution were actually sugars and amino acids that are used by butterflies. So we built 10 prototype of camera trap, and we were doing the test in 2 different sites. And hopefully it was working. So butterflies were coming under the trap, and they laid or flying around the different treatments, and they use their proboscis to suck the nectar from the wick. We collected during this year from May to September more than 350,000 pictures and videos. So every time we collected a video of 20 seconds where we extracted a picture. So we run roughly an algorithm to detect how many pictures from butterflies we got. It was around 28,000 pictures. And of course, what we were interested was to relate the presence of butterfly or other species to the treatment we had on the plate. And for this, we had no other solution to do it manually. So it was too many times. It was really time consuming. So we had a student that would only take the pictures from May to June and to watch every video and associated the species when the species was flying around the treatment or sucking the wicket. So we did that for butterflies, but for other species, so we used 2,000 pictures for that. And the first results were quite interesting. So we can see that actually on the graph A, most of the species coming under the trap. This is not good. Actually, more bees were coming under the trap than butterflies, but the second number of species coming were the Rhophalocera, so the pink colors here, so the day butterflies. Then we had also other insects in green, Coleoptera and whatever, and other Lepidoptera, mostly moth. Maybe it could be some confusion sometimes with some small Rhophalocera. So it was working quite well. And we show on the graph B that the Buglea solution, so the Buglea treatment, it's the middle one, was the most attractive solution for butterflies. So butterflies is in pink. So we collected more data with the Buglea solution than the other one. And also, which was interesting, sorry, because the quality is bad, but With the white cap, with the white color, we collected less data for butterflies. So apparently the white color was less attractive for butterflies. And lastly, interestingly, when we added or not the nutritive solution, so sugar and amino acid, on this slide, you can see that when we added the sugar, we had a lot of noise coming from bees. Of course, they learn. So which means that if we want to avoid too much noise for the bees, if it's not the goal of the study, it would be more interesting to probably remove sugar. And in that case, when we remove sugar, we can see that we have much less bees. And there was a difference in butterflies, but not so much. So butterflies were also still coming to the attractant, even if the sugar was not added into the solution. So I will be very quick in this one, but we compare the data between traditional approaches because we have IBMS transect going at the same place, exactly the same place as where we put it, the camera trap. And so we had 8 passages, and we actually see that only for those days where we went on the transect, we actually detected more species with the camera trap than with the traditional approach when we worked one time the transect, which is also very encouraging results. So why I'm here, it's because we want to continue and develop a new project with the idea of using this attractant with camera trap. And of course, those projects involve a lot of different competencies, as you can understand. So for the field experiment, we need an hardware. We need also to work on the chemistry side. We also would like to have a nice software that will help us to analyze all the pictures that we are collecting, also with the possibilities to associate determination of the species to a treatment if we have several treatments under the camera trap. And also, of course, it involves the interpretation of the results, which is ecological research, because starting from that side, we are very interested and we can work on that. Because, of course, what will be the consequence of using an attractant on the detection rates can be different than if we don't use an attractant, of course. But can we still use the proxy of abundance of species? What would be the species more attracted than the others, and so on? All these questions are related to the behavior of the butterflies and other pollinators. And so there is a lot of things going on in the pollination ecology. And I know that my former PhD supervisor, Hans van Dijk from UCL, is very interested also by those questions, so we can team up, for instance, for that. We are also strong into the chemistry side because we can make all the molecule extraction, characterization, and so on, and test some stuff on the lab. But of course, we would like to team with you guys for the hardware side. We know that a lot of teams have more evolved hardware, and also through the software side. So please get in touch with me today, tomorrow, by emails, because we would like to try to team up and write a project and continue investigating all the different possibilities we can have using attractant with camera trap. Thank you very much.
Thank you.